Hi guys, Dr. Douglas Gillard here once again. We're going to do another embryology video. We're going to start right from the beginning. I didn't like the other one. I've actually got a few more books on the subject. I actually got the class now, so that was kind of strange. I was just doing it to increase my skill in the area. Ended up getting the class for embryology, so all this work was not in vain. So we're going to talk about fertilization through the formation of the very early embryo in this video. All right, so that's where we're going. This, if I could only pick one book, now this is a close call now. It used to be Larson. Larson, I think, is still a little more readable, but I am really starting to like a book called Carlson. It is by far the deepest book, and if you want to understand the entire story, this is probably the book to get. Some of it's not quite as good of a storyteller, but if you really want to understand embryology, you can watch my videos, or you can buy all these books and read them all like I did, and you can piece together a pretty darn good story. Okay, there's Carlson, my number 2 pick at this time. And I would almost say if I had to give up one book now, I can't give this book up. I just keep coming back to it. So this is a really good book. All right, here we go, fertilization. So where are we going? The big picture. The fertilization involves the meeting of the male and female gametes. The female gamete, of course, is the egg. Here's a cartoon of an egg. I got to give a plug to this video too. And there is the sperm, and the two gametes meet and they mingle genetic material, and we have ourselves a tiny little human called a zygote. And we'll plug out to this. I'm actually going to make my students watch this video before anything. This is it's not, he doesn't attach all the correct words and things like that, but it's a very costly video, and it's just incredible. This actually came from it as a screenshot. So you just have to watch this. If you watch this video, I would stop this right now and watch this, and you'll get an idea of where we're going, and then come back here. So in general, as I just said, fertilization is the union of the male and female gametes. Specifically, the union is of their nuclei, or that's what actually melts together. This fertilization process typically occurs in the ampulla of the fallopian tube, and it has a span. Fertilization starts, now arguably, Moore, which is a Board of Chiropractic Examiner book, and Carlson, they both say it starts when the sperm penetrates the very outer layer of the egg. And that's called the Zona pellucida. You can also, by the way, I'm going to use this interchangeably, but oocyte is another word for egg. They're both AKAs. And it continues, and it officially ends when the two nuclei of the maternal, or the two nuclei of the sperm and egg melt together, and all the chromosomes mingle together. That's when it ends. That, after fertilization is over, that is day 0 in the clock that we will talk quite a bit about. Okay, so right now we're actually below ground 0. Here's kind of where we're going in general. So here's the secondary oocyte, and that's not pronounced oocyte or oocyte. Some people call it oocyte, but oocyte, if that could be changed to an A, would be correct. Or you can just call it an egg. So an egg and a sperm meet up together. Fertilization is ground 0, and we're going to go all the way here through the creation of the zygote. Actually, we're going to go right to here, to 2-cell zygote. Your anatomy, basic anatomy, here's a fallopian tube. Of course, you have the isthmus, is this little part right here. The ampulla would start right about here and run to about here. Classically, professors will pin it, or this bulging region is where the fertilization usually occurs, but technically this is all ampulla. The infundibulum is this very end portion, and that connects to the fimbriae. These are little fingers that gather the egg and sweep it inside the tube. The ovary is not shown, but the ovary would be right here. That ovary is what produces the egg. Let's meet the sperm. So the sperm has some pieces. It's kind of a cool little creature. It has a tail that wiggles like crazy, and it literally swims. Its job is to swim up through the vagina into the uterus and get into the fallopian tubes and find that egg. It also has this middle connecting piece here, and then it has a head. We'll look at that here more in a second. The head is really what we're going to talk about because it has some components. Its plasma membrane is this very outer region. Let's actually blow this up. Here's the plasma membrane all cells have a phospholipid bilayer plasma membrane, and that's it right there. Then underneath it, it has something called the acrosome. Looks like a little football helmet kind of. And then underneath that, this is the nucleus of this cell. So we have a nuclear envelope or a nuclear membrane, this inner layer. So these are all important. This is what's going to eventually have to break down. The DNA and chromosomes are all contained within the nucleus, which is here. So we'll get to that. So that's the sperm. The sperms, that's general facts. You guys already know this. It's created in the testes by Leydig cells. It's released goes up the vas deferens to the back of the prostate. The vas deferens widens out here into a little reservoir called the ampulla. The ampulla lays on the back of the seminal vesicles. Both of these have fluid that is injected through these ejaculatory ducts right into the center of this big thing. We just covered this actually in GIGU, my pathology class there. That's the prostate gland. It's injected into the prosthetic portion of the urethra. And the sperm, of course, is not alone. It joins the prosthetic and the seminal juices, all of which keep the sperm alive and keep it in a gel form until it gets into the vagina. And then actually the prostate juice, actually PSA, is what breaks that gel down and releases the sperm so they can swim like crazy and try to get up and find the egg. Important receptors. So on the plasma membrane that we just showed you, there are some important binding and docking receptors. These are the SED1 receptors, SED1 receptors, or they're sometimes just called SED1 proteins. But their job is to bind the Zona pellucida, and we'll get to that here in a minute. There are some receptors that meet these ZP3 receptors that these two hook to, and we'll see that here in a minute. On average, there's about 130 million sperm that are ejaculated during intercourse, and they're ejaculated. Well, they can be ejaculated anywhere, right? But if they're ejaculated, the goal if you're trying to have a baby, they have to be ejaculated into the vagina, and a lot of them die off. The vagina is quite acidic, so tens of thousands of them die off in the vagina. But they're all swimming as hard. Only the strongest ones and luckiest ones will make it all the way up into the fallopian tubes. And again, about 1%, a few hundred of them will actually make it into the correct ampulla region and fertilization. Some of them go up the other tube, but remember the egg is only injected into, you have your one cycle, it's injected into the left fallopian tube. The next cycle, it's into the right fallopian tube, so it alternates like that. There's some new research out that shows that there appears to be a sperm chemotrophic factor that's released on the surface of the oocyte, and the sperm has little olfactory receptors that can sniff. I mean, it's not really like a bloodhound, but that's the idea. They literally can sense or smell this factor, and it helps them zero in on that egg. Even if the egg, for whatever reason, if the fimbriae don't gather the egg and the egg falls into the pelvic cavity inside the peritoneal cavity, the sperm have been known to swim right out of the fallopian tube and go into the peritoneal cavity and impregnate and fertilize the egg while it's floating around in the pelvic cavity, which is an ectopic pregnancy and is very dangerous. You can't have a baby like that. Doesn't happen very often, but that's not a good thing. Okay, let's talk about the egg or the oocyte. There's that same video I showed you at the beginning of this video that you should really go watch that. That's kind of what it looks like. That's the Zona pellucida we're looking at there. That's 2-3 layers thick. The oocyte, it's released from the ovary, of course, during ovulation, gathered by the fembria, swept into the infundibulum. Interesting about this egg, now you remember your genetics, the gametes go through meiosis to form haploid gametes. So the egg is actually hasn't even gone all the way through meiosis. It's actually stuck in metaphase 2 of meiosis. Meiosis, meiosis, tomatoes, however you want to say that. I've always learned it as meiosis. So that's interesting. When the sperm catch up with this egg, it actually stimulates the nucleus of the egg to kick into gear, and it'll finally complete meiosis. So that's kind of strange. There are 3 key layers here that the sperm has to get through. There's the corona radiata, zona pellucida, and the plasma membrane. Some authors call it the vitalin membrane, but I'm just going to call it the plasma membrane because that's what it is. Here's a really nice drawing of a oocyte, or the egg. And we can see, now they didn't draw this. Sometimes this is usually 3 layers thick, but sometimes it's 1, sometimes it's 2. But this is the corona radiata around the outside. Then we run into the zona pellucida here. Okay, and there's a little space here. There's 2 little spaces we'll look at. And then we get to the actual plasma membrane of the cell, would be this end. Sperm's got to get through all 3 of those, including there's 2 spaces it's got to. The spaces are easy to get through, but it's not so easy to get through the zona pellucida. How do the sperm get through the barriers? The sperm, there's 2 processes that need to go on. In fact, the sperm is not even a real complete sperm until it undergoes something called capacitation. And then it's going to undergo an acrosome reaction too. But all sperm have to be capacitated to be real mature sperm. So we'll take a look at these processes. In fact, capacitation is first up. It's final step in sperm maturation. It's started by the sperm contacting the oviduct. What the heck is the oviduct? That is just the inside lining, the inside hole of the fallopian tube, or it's the lumen of the fallopian tube. So when the sperm start bumping into that, something is released, the unknown factor is released, and that starts the process of capacitation. And we go to Carlson to get the full story here. There's 2 phases. In phase 1, the sperm's plasma membrane kicks loose cholesterol. Cholesterol ends up blocking the capacitation process. If guys don't have cholesterol around the plasma membrane of their sperm, they cannot get somebody pregnant. So cholesterol is very important because they'll be capacitated the minute they come out. So cholesterol is important. In phase 2, this acrosome is going to lose glycoproteins. We won't get any deeper than that. We'll just say it loses glycoproteins. And now it's capacitated and it's ready to start things like the acrosomal reaction, which is very important. Another thing capacitation does, you can't, like if you look at a sperm, you can't tell whether it's capacitated or not, a picture of a sperm. But if you look at a live sperm, it's crazy hyperactive. So it really kicks the flagella into motion, and that sperm is swimming like crazy. So that's a way you can tell capacitated sperm. Increased tail movement, which is going to help power through the corona radiata and the zona pellucida, those outer layers of the egg. Corona radiata, it's just like the corona of a sun. There's an eclipse, a solar eclipse, and we can see it's kind of similar. Only that's not, of course, cells, but you get the point there. So it's the very outer layer around the oocyte. It's highly cellular, so 3 layers thick usually, and the intercellular matrix between the cells is very thick with hyaluronic acid. So normal sperm couldn't go through there, but a capacitated sperm, we'll see what happens, can get through there. So that's the first barrier that must be conquered. So how do you get through the corona radiata? The corona radiata can secrete hyaluronidase, and that, of course, can cut right through the hyaluronic acid-rich matrix. And that's how it does it. And remember, the sperm is hyperactive, so there you can't see the heads of the sperm, but those are the tails. Those things are fluttering like crazy, and it's pushing its way between the cells of the corona radiata. Now we got the next obstacle is the zona pellucida. This one's not as easy because there's no cells to push through. So it's fairly clear layer. It's deep to the corona radiata. It's made up of two things: glycoproteins called zona pellucida sperm binding proteins, or just zona pellucida glycoproteins, and glycosaminoglycans. We're not going to talk about those too much because they're not super important to us, but these are especially, and those are abbreviated to ZP proteins or glycoproteins. ZP3 is kind of the star of the show, as we'll see. This protein acts as a receptor, and that's what binds the SED1 receptor that we just talked about of the sperm. So the sperm plasma membrane has SED1 receptors, and it's looking for these ZP3 receptors on the zona pellucida. Here is a cartoon, a very deep cartoon. There's the zona pellucida. Here's the corona radiata. Here's the cytosol, or the cytoplasm of the oocyte. So the sperm's job is to get through here and get in here and meet up with the nucleus of the oocyte. So it's already made its way through the corona radiata, and now it's docking. There's its SED1 proteins, which are magnified, of course. They're not that big, but this is just so you remember it. And they bind to this little blue ball here, and that's the ZP3 protein that acts as a receptor. So you got to have, all of these have to be functioning, or you're going to be sterile. If the guys if they have mutated genes for SED1 receptors, they make a mutated SED1 receptor, and it can't bind to the ZP3 proteins, you're going to be infertile. Same if the female has mutated genes for ZP3 and the male's okay, but you can't bind again. So the female's got to have normal gene product. These ZP3 proteins have to be normal. Right? So getting through, just to recap again, the first thing, or getting through the zona pellucida, you can kind of guess already. First thing that happens is the sperm's SED1 receptors bind to the zona pellucida's ZP3 receptors. Assuming they're both normal, they will dock real nicely, and the sperm's head will be attached to the zona pellucida. As we said, mutations in any of these results in infertility. So this binding stimulates, here's a board word, you got to know this word. This binding stimulates what's called the acrosomal reaction, or the acrosome reaction, or acrosomal reaction. So that's very important. What is that? This binding stimulates the release of some pretty strong enzymes, especially, and these come out of the sperm, or out of the acrosome. They're released. We won't get into that mechanism, but they're spit right out. And they're like acid. They eat themselves right through the zona pellucida. The king of these is called acrosome, the most important one by far. It's a serine protease. Protease eats protein, so it can literally eat right through the zona pellucida. There's a pepsin-like substance, Carlson. The chiropractic board books didn't mention this one. Moore, which is a chiropractic board book, mentioned esterase, and this neuraminidase is other ones. But the key one, again, is one you should put in your noodles is acrosome. If you have a mutation in the gene that makes acrosome, guess what? You can't have babies. So it's another thing that can go wrong in guys who are infertile. All right, so these enzymes are all released together, and they cause a focal lysis, which is a nice way of saying they rip a hole in the zona pellucida, and now the sperm is in. Here's the zona pellucida. We're heading this way. Here's the oocyte's plasma membrane. This sperm has already ripped its way through the zona pellucida because of the release of those enzymes, and now it's heading for the plasma membrane as the next target. There's a little space here that kind of the eye of the hurricane, I guess. This is called the perivitalin space. Not really much, or at least we don't know of anything that really happens in here other than the sperm has to swim through there. So once the sperm is through this layer, fertilization, according to Moore, Chiropractic Board Book, fertilization has officially begun. So, blast the horn. Fertilization has now begun. It's not over yet, but it has begun because we have penetrated the zona pellucida. Got it? All right, so there's that perivitalin space. Now the sperm has bound to all these little things sticking up. So those are microvilli. So the plasma membrane has microvilli, just like the microvilli of the intestine, kind of. They don't have cilia, but it's the same kind of idea. So after a slight delay, sperm swims through the perivitalin space, binds with the plasma membrane. Specifically, sperm binds to the microvilla, as we just said. Some fun facts. These are relatively new. I'm not sure if these will be on boards. These are from Carlson, though, which is not a board book, but it's a great deep book. There are two plasma membrane proteins thought to be involved. There's one called izumo that lives on the sperm plasma membrane, and there's one called CD9 that lives on the oocyte plasma membrane, and these two bind together, and after that the sperm kind of gets engulfed by the plasma membrane and swallowed, and the sperm is swallowed inside the plasma membrane of the oocyte. The oocyte, the first polar body, remember the egg was stuck in metaphase 2, but the metaphase 1 gave us, or the completion of meiosis 1 gave us a polar body. We don't want that to be fertilized, right? Because that has chromosomes. So this actually doesn't have microvilli over the top of it. So the sperm can't bind above the first polar body, so they can't fertilize it, which would not take. You can't get a baby like that. You'd get a miscarriage. Just another nice picture here. So here's the hyaluronidase being released by this sperm head. So this one's trying to go through the corona radiata. This one is already bound to the zona pellucida and released its enzymes and cut a hole through there. But unfortunately, this sperm beat them all to the punch, and the membranes, the plasma membranes of the sperm and oocyte have bound together, and the inside of the sperm has actually gotten into the target. So the game is successful. This sperm is the winner, and we'll see a reaction that occurs. All these sperm are going to be shut out here in a second. But this is fertilization. Okay, entering the cytosol. So just kind of what I said, almost the entire sperm is taken in except for its plasma membrane. So it's got, for whatever reason, these two plasma membranes bind together such that this one can't get in. And this wouldn't be sticking out like this. This would be laying flat like we saw in that picture. But nevertheless, only the inner part gets in. The most important, and sometimes even the tail doesn't get. The only part we need to get in is the nucleus, or underneath the acrosome here. Okay, so everything's taken in except for the plasma membrane. I think we got this covered, but there's something important here. So the presence of the sperm within the cytosol, and we don't know the reaction completely. I'm not even going to go down the pathway, but it triggers some very important reactions, and let's talk about those. Just looked at that already. And a good way to talk about it is talking about polyspermia, because polyspermy is triplets, quadruplets, quintuplets. You can have, if more than one sperm gets in here, like say this one gets in here, it's possible to have two embryos created. So we don't want that. I mean, birth is dangerous. I mean, back in nature, birth is much more dangerous. The female, nowadays it's not that big a deal. You could do a C-section. Back in the day in nature, if you didn't have that, it'd be dangerous. So the body tries to prevent polyspermia. So there's two blocks. There's a fast and slow block. So let's talk about, this is from Carlson, fast block polyspermia. So here's what happens when the sperm finally gets into the cytosol of the oocyte. A huge depolarization of the cell membrane occurs. The cell membrane, semi to other cells in the body, is about negative 70 millivolts. And the second the sperm enters the oocyte's cytosol, it jumps way up to plus 10 millivolts. The sperm need this negativity to get through plasma membrane, so it prevents the binding of other spermatozoa to the plasma membrane because of this change in voltage. That's one mechanism. So one way to stop. There's more though. So now we go to slow block polyspermy mechanisms. So the second the sperm gets in the cytosol, and this is an important one, these waves of calcium are released from organelles in the cell, and you get these surges of calcium sweeping over the egg. So those are important. First of all, those start second meiosis up again. Remember, we were stuck in meiosis 2, and that is started. So this takes off from metaphase 2, heads to anaphase, telophase, you know the deal. But we're in meiosis, we're not in mitosis yet. So it finishes off the maturation. The egg is not even fully mature at this point. But the second the sperm gets in there, it's going to be mature really, really quick. That wave of calcium also stimulates the cortical granules, which float around in the cytosol of the egg. And it stimulates them to go up and dock to the plasma membrane and spit their contents into that perivitalin space. So we'll see what happens to that in a second. There's also causes a decondensation of the nuclei, which we'll talk about later. That's really just part of a mitosis that's going to occur. Okay, so those are the three things. Right, cortical granules deserves a little bit more talking. So we said they are floating around, and the wave of calcium stimulates them to bind to the cell membrane and spit their contents into that space. Those contents include proteolytic enzymes and polysaccharides. Polysaccharides, we won't get into all the different types of them. Bottom line is, those ones that are spit out, they bind to the zona pellucida and cause it to swell up and get stiff. Now it's almost impossible for sperm to get through there. Sperm that are caught halfway through the zona pellucida will be crushed as this thing swells up. So that's another mechanism of preventing sperm to get through the zona pellucida and get into the oocyte. This swelling up and stiffening up has a name. That's called the Zona reaction. There's another thing to put into your noodles, a good board question. So that's the Zona reaction. Its Zona reaction is the releasing of proteolytic and polysaccharides secondary to that wave of calcium, and they are released right into this space. Where'd it go? Right here, this perivitalin space. Here's the zona pellucida, so they're spit out right into this area. They come out of here. There's little, I guess I should get a picture of this one day, but they're little vesicles, and they bind to this and spit their glycoproteins out here. Those bind with the zona pellucida, the backside, and it causes this zona pellucida to get super stiff, super hard, and sperm can't get through it anymore. Okay, cortical granules. So that was the Zona reaction is caused from the polysaccharides. What about the proteolytic enzymes? What do they do? Here's a third way that polyspermia is prevented. So the proteolytic enzymes from these cortical granules, they bind to the ZP3 receptors on the zona pellucida, and they destroy them. So now sperm can't, if they do manage to get that far, they can't bind to the zona pellucida anymore. Okay, so it's because there's no more ZP3 proteins. So yet another mechanism of sperm, sperm binding. So bottom line is extremely difficult for more than one sperm to get into the oocyte. Okay, now we already said this, that when the lucky sperm does get into the oocyte, it stimulates a wave of calcium to sweep through the oocyte, and that stimulates meiosis 2 to kick into gear. And so that gives us finally a mature oocyte with a normal mature nucleus. But remember, just like the sperm's nucleus, these are haploid. And go back to your genetics class. I'm not going to go too deep into that, but they're haploid, meaning they have half the number of chromosomes that you have in all the cells of your body. Scrape your cheek cells. You probably did this in high school these days. Scrape your cheek cell, and that, well, you probably didn't do this in high school, but you scrape your cheek cell, or take cells from your fingertip or your nose or anywhere, and you look at those cells. They have 46 chromosomes in them. 44 really in 2 sex chromosomes, but 46 in total. And those are diploid, those are 2N. But these guys are 1N. But anyway, the wave of calcium makes it finish off. It's in anaphase now. And go back to your basic cell biology if you don't know what I'm talking about. But anaphase is where the chromosomes have lined up and they've pulled apart. And you can see them going to their respective poles. So this is going to split, and it's not going to create 2 nucleuses, right? It's going to create the secondary polar body and then the nucleus, which we need, the mature nucleus. Okay. At this point, once it's finished meiosis 2, we have ourselves a real mature oocyte, and that can be called the definitive oocyte. Pretty good? All right, and here's a cartoon of finishing up meiosis. So here's anaphase. The chromatids have been ripped apart. They're being pulled by these spindle fibers to the poles here. And the cytokinesis is going to occur and wall this off. But we're not going to make 2. We don't need 2 nuclei, but we're going to make our secondary polar body, which kind of dries up over here. The real nuclei will be right here. But the key is here, these are considered haploid cells. Did I say diploid? Which gives us, 1N nucleus. And so each one of these that's 1N and that's 1N. They have half the number of chromosomes, but this one's going to be disintegrated. Okay, now pronucleus. What the heck's that? So pronucleus is something we can see under the microscope. The chromosome material gets really, really thick. And let's talk about this weirdness. So we have a really weird type of cell now, right? So we have the sperm. The sperm head contains the nucleus. So we have the nucleus of the male gamete, the sperm, and we have the nucleus of the female gamete, the oocyte, in the same darn cell. So we got this weird cell with 2 nuclei, 2 polar bodies too, but they're all degenerated by now. But we have 2 sets of 2 nuclei, each carrying a haploid number of chromosomes. And you can figure out immediately how we're going to make this right and get back to diploid. 46 chromosomes, we're going to mix these 2 together. And these 2 have already come together in this picture. And this cell, part of the cell membrane, is going to disappear. And these guys are all going to mix together. And that's exactly what happens. The nuclei are now pulled together by these really thin microtubules that you can't see. There's some more microtubules that are going to be connecting in metaphase in a second. Okay, but the 2 nuclei are moving together. And now we're done with meiosis, or meiosis is done. But this, once these come together, these guys are going to switch and go through regular old mitosis. And we're going to divide this one cell that's going to occur here in a second. I'm getting too far ahead of my slides. So 2 nuclei move together. They're both going to go through standard mitosis. So as these guys are moving toward each other, these started way over here. The male pronucleus was over here. They're moving toward, but even as they're doing that, we've started interphase, and the chromosome material is replicating. Then we're going to prophase, and the chromosomes are starting to get thick and visible. Then metaphase, they're going to line up here in a second. So that's regular mitosis is going on. But when the pronucleus, when you're able to see these thick this thickening of the nuclei, indicating they're in mitosis. They've went through interphase. That they're no longer called nuclei. They're called pronuclei. So pronuclei is a special term. Okay, that means they are in mitosis. Okay, everything I said. So they went through interphase. They're replicating. They each made copies of the chromosomes. So now there's not one linear chromosome. There's two chromatids which are held together by a centromere. We'll look at that. That's your basic genetic stuff that hopefully you've had classes, or cell biology. We'll go through that as well. So next we go into prophase. For our weird little cell is going to go into prophase. So here's so this is already went through interphase. We have a centromere here holding together the two. Maybe this was the original strand right here, and this one was replicated. But nevertheless, we have two chromatids. But this is still, people get confused here. Even though really it's going to become two DNA molecules in a second when this is ripped apart in anaphase, right now it's still considered one chromosome. It just has two pairs of DNA, or two chromatids. But it's still counted, the cell is still 1N until we get anaphase going here. This is always a little confusing part. So here's the pronuclei again. I guess the slide's one out of place, but you can see the sperm tail is degenerating, withering away, but the nuclei are going to be moving. The pronuclei are moving toward each other at this point. Here's a real picture of a human pronuclei that have pulled right together. Pretty soon this membrane's going to fuse, and this is going to come apart. So, but now things get weird. Things have already been weird, but now they're going to get weirder. So this is going to act like one nucleus now. This membrane's going to disappear, and we're going to run through mitosis, not meiosis. So metaphase and anaphase will not happen in the individual nuclei. So you're not going to have that going on. It's going to happen in both of these coming together. I know that's kind of confusing. But I mean, interphase and prophase happened in each of these, but metaphase, which is the next phase, is not going to occur in both of these at the same time. It's going to wait for a second till these form one unit, and then metaphase is going to occur in the entire unit, and then telophase will occur in the whole unit. So it's kind of weird the way the books don't even really describe this. That's from my biology background. I know this, but that's what happens. So instead, the nuclear membranes have to bind together, or come together into one big happy nuclei, and that's what happens. So here is sometimes the picture's better, right? So here's our two pronuclei. They've bound together. The membranes between them have been dissolved, and hey, we got it's a little weird looking, but now we're like a normal cell. We got one nucleus in the middle. The genetic material from both gametes is now going to mix together. And yep, male and females are going to come together, and fertilization is now completed. So come together. There's the Beatles, right? Great album if you've never experienced the Beatles. But yep, meet the zygote. The zygote is born. So most authors agree at this point, when you get here, when the genetic material mixes together, now you not only have completed fertilization, but that little celled creature has a name. It's now called the zygote. The zygote. So very important slide. The Beatles slide is very important. So now you have just created, it's no longer haploid, right? This was 1N. This was 1N, 23 chromosomes, 23 chromosomes. What's 23 plus 23? 46. Hey, we just went to diploid. Now that's just our designation, but it's now a diploid 46-cell chromosomed creature. And so great. We're like a normal human now. Okay? Everybody get that? What's the results of fertilization? So this is always an important slide. You can see test questions coming from this. So it stimulated the female gamete to complete its second meiotic division, right? We talked about that. So it basically, at the end of it, it restores a normal diploid number of 46 chromosomes because those pronuclei melted together. The chromosomes have also mixed together. There's crossover that happens we didn't talk about. That's genetics and cell biology 101. But you have a crossover occurs and you get a mixing of mom and dad. So the offspring or the baby is not going to come out like a clone of mom or a clone of dad. It's going to be unique because of the mixing of mom and dad's chromosomes. So everybody is unique because of this. It also determines the sex of the baby because the sperm can only give an X chromosome or a Y chromosome. The female can only give X chromosome. So if it happens to be a Y carrying sperm, you're going to have yourself a male zygote. If it's an X carrying sperm and that melts with the female, in that I'm talking about when the two pronuclei come together and all the chromosomes mixed together, maybe the sperm that got in there was an X chromosome, you're going to have yourself a female. Okay, more basic genetics. So it's also going to initiate the phenomenon of cleavage, which is really just telophase, right, or telophase. Tomatoes, telophase. So the process of mitosis is now going on. We're currently kind of stuck in prophase to say, okay, fertilization is done. The zygote is born. Great, we're in prophase, but we're going to keep right on going. And that's going to get into the next stage, which is called cleavage. So cleavage begins. I was going to make this a separate video, but because we're talking about mitosis, let's just keep rolling with this a little bit. So what about metaphase? We stopped in prophase. And so what about that anaphase and telophase? We haven't finished that. Well, we're going to keep right on going. Just because fertilization is done and we created the zygote, we keep right on rolling. And so metaphase kicks into gear at this point. So metaphase, as we know from basic biology, the chromosomes will line up kind of on the equator. Microtubules will form and attach to the centrosome or centrosomes. The centrosomes are like little fishing reels. They're going to reel in the chromosomes. They attach to the centromeres. And we've already went through interphase. We're starting metaphase. Remember, these are still, they're double. They've already been replicated. So we have chromatids now, just like before with a centromere holding them together. I think I have a picture. Do I have a picture? Yeah, I do have a picture. So here's where we are. So we're starting the cleavage phase now of embryogenesis. And here we are. There's the centromere. Say this was the parent, just like we did before with meiosis, but we're in mitosis. There's the parent. So in interphase, we've created a new clone of that. And these two, can't call them chromosomes yet. They're just one chromosome because they're still bound together by the centromere. And there's some more. The centromere, it's really a centromeric zone. There's deeper in cell biology, it has more parts. But for us, I think centromere is fine. And we got these microtubules that have connected centrosomes, which also replicated in interphase, are over here. These are the fishing reels. And they're literally going to pull these apart when we get an anaphase. This one's going to start reeling this fish in. This one's going to reel this fish in. And this is going to split in anaphase here in a second. Anything else I need to say? But these are still considered one chromosome. Okay, are we good? So now what about anaphase? Now the fishing reels have kicked in and we have split those chromosomes in half. So that's what anaphase does. It rips. Anaphase pulls these guys. The centromeres break, become centrioles at that part. 2 centrioles equals a centromere. We're in anaphase. The chromosomes are pulled apart. They're going to be pulled over to their opposite poles. Because what's the goal here? We can't have a human with one cell. We got to have a human with trillions of cells. So we need to start splitting this thing like crazy. And that's what we're doing. We're creating 2 new cells here. So let's pull these chromosomes to this half, pull these chromosomes to this half. And now this is the phase where technically the human or the zygote has 92 chromosomes because now the chromatids have been split And so you can say that in anaphase, you actually do have 92 chromosomes. Great biology test question, right? I'm sure you know that already, but if you don't, now we still call it 2N though. That gets, why don't you call it 4N? They still call it diploid. This is not going to last long because we're going to wall this off, and each side is going to take 46 chromosomes. But for a brief second here, or for a few hours anyway, we have a 92-celled chromosome creature. Okay, everybody good? We're done with anaphase. Now what about telophase or telophase? Telophase is going to occur. This is the final phase. We're going to create 2 new daughter cells. We're just going to expand on this, but now we need to cave in the cell membrane here and split this in half, and that's exactly what we're going to do. Sister chromatids are they're reeled in with a fishing reel all the way over. Nuclear membrane is then going to form. So if you get these all the way over here, we're going to form another nuclear membrane around this. We're going to form another nuclear membrane around this batch because we're going to have 2 separate cells. They need their own nuclear membranes. And then a cleavage plane or furrow develops, and then cytokinesis occurs. This is the splitting in half of the cell. I think I got a picture. So here's the cleavage plane develops, and you can see a rip or a tear going right here, or a wall built right here, and that's called cytokinesis. The cytoplasm is divided. We have 2 cells, so hey, our 2-celled zygote has got closer to a human, because now our 1-celled zygote has got closer to a human because it's 2 cells. So we're going to keep repeating this process until we have trillions of cells made. These are no longer, like probably in biology you've learned, these are called daughter cells, and they're still daughter cells, but we don't call them that anymore. We call them blastomeres. This mitosis process has resulted in 2 blastomeres. Chromosomes are now getting crazy and wild again, and we're gonna run through mitosis again with both of these. So pretty soon we're going to have a 4-celled creature. So meet the embryo. At this part, we met the zygote. Now we're going to meet the embryo. Once we have 2 blastomeres or 2 daughter cells like this, we give it a new name. We call it the embryo. Most authors, not all, agree with that. Some say this right here is the beginning of life, where our 1-celled creature split into our 2-celled creature. Okay, so now we have an ast. And if you can't remember, you can still refer to this as a zygote as well. But officially it's an embryo. So here's another picture of this. There's our 2-celled zygote, or embryo, if you will. Some say, like Larson, that life has begun right here. There's a real picture of a human embryo, 2-celled stage, 2 blastomeres. All right, we'll leave it right there. I'll do another. We'll keep on going. We're going to form a marula next, a little mulberry, and we're going to keep right on rolling with this stuff. Okay, hope you enjoyed it. Please give me a thumbs up. Subscribe to my page if you like this kind of stuff, or my channel, not my page. You can check out my website as well, chirogeek.com. And we'll see you in the next one.